What I was asked to do is to just describe how we go about financing new stuff, particularly stuff at the leading edge of something that hasn't been done before, unprecedented. So the first question you have to ask with innovation is why bother, right? And it sounds like a dumb question, but it isn't. Just because it's trendy doesn't mean it's the right thing to do. And you know, if you put yourself in a venture capitalist's shoes, if you hear the words game changer or radical, then what they hear is high risk, probably won't work, right? And for a large part, they're right. So from a financial perspective, innovation is not necessarily good news. In fact, the ideal is to be just slightly behind that innovation wave. So let somebody else do the hard yards to work out whether this thing's really going to work and where it's going to work, and then come in with something better after that. It's way better from a financial perspective to come in at that point. So the real race is usually first to be second. Very low technical risk, it's got a clear development path. You know that differentiation is basically what you will live or die by, but you can deal with that. And you don't have to say which clinical indication is really sweet for this particular mode. You already know. You've just got to come up with something better in that same framework. So it's much less diffuse, and the technical risk is lower. And when you come on to move that program on, then other people can join the dots because they can see what the primary compound looks like or the primary approach looks like, and then they can join the dots from where they are. Right? Much simpler, much cleaner. But we live as part of an ecosystem. I mean, this is about partnerships and everybody working with different disciplines and trying to integrate that. It actually goes beyond that. The whole thing is an ecosystem. And we're just one part of it. So we, being the venture capitalists, are just part of that ecosystem, where the challenge is not to get regulatory approval, and I'm sorry, I know a lot of you work on getting regulatory approval, but that's actually not the real challenge. The real challenge is getting paid for that. So reimbursement is the issue. That's the challenge that certainly the pharma companies now feel most acutely, I think. And because they do it, and that's their pain point, it's also ours. Because if something really isn't, or is a little bit iffy about getting reimbursement, the chances are they won't go there. And then we'll be stuck with whatever company we've got in our portfolio. So the reality is we're financing innovation because we don't really have a choice. It's actually more fun, but we really don't have a choice. So the thing about venture, and I've come at it from the operational side of things. So I've basically spent most of my lifetime as an entrepreneur, and now I'm working with a venture firm, but still practicing that trade. And the really interesting thing is that for a venture capital firm, there are really only two constituencies they're answerable to. One of them is the LPs, the limited partners, the investors in the fund that you're deploying. These are insurance companies, pension funds, things like that, and the Financial Services Authority. Beyond those two constituencies, you can do anything. Anything goes. And in fact, when I first started hanging out with Index, we were talking over lunch, and I'd been working in a medium-sized biotech, and I mentioned that actually it was much easier for me to manage projects virtually than to try and get them done internally. So rather than having a daily battle for resource, timing on developing these programs, I actually just went out to CROs and deployed the budget externally. And it was actually a recipe for a happy life as far as I was concerned. And I thought that we were doing biotech companies the wrong way. We should actually just be having these small virtual SWAT teams working with external expertise without having to build the infrastructure ourselves. And this is what we call asset-based financing. And 10 years ago, we pioneered the use of asset-based financing, or the approach of asset-based financing. Now it's becoming more and more the norm, because one of the consequences of that is that it's much more capital efficient. So you buy programs much more cheaply, but you accept that most things are going to fail. And what you really have to do, if you're working on really innovative stuff, a lot of this stuff will fail. You just have to accept that and develop a strategy that will tolerate it. Because failure as a business approach is rarely a good one. So you've really got to be able to control that. And one of the concepts that we've come across, or that we've put in place, is one we call a killer experiment. So rather than trying to keep programs alive, we actively try to kill them. So, and we do this right from the start, and we do it in a stepwise fashion. And the way we do that is with a part of a fund called the seed pool. Now, most venture firms have something called a seed pool, which is something for financing very early stage programs. And they've been part of the fabric of the venture capital industry for decades. But they tend not to be used strategically. They're used when, if and when they're needed. If you just happen to see a very early stage thing, you just want to put a bit of money in it to get it off the ground. It's part of the day-to-day activity. What we've done is we've turned it now into a strategic tool. And basically, what this means for us is that we can put anything up to $1 million in a project, and in aggregate up to 10% of the fund. But it's something that we can write off, right? It's much lighter touch for us. It's much easier for us to get this done, much quicker, and we don't have such a high expectation. And what that does is it changes the behavioral dynamic. Because if you've got $1 million at risk that you can afford to lose, 10% of the fund is not going to be material in the final analysis. So you can afford to lose this. If you've got something like that, it just makes you much more able to tolerate risk. So you can do some really crazy stuff if you're not really worried about failing. And because you've only got $1 million maximum and 10% aggregate on these sort of ideas, then you really anything goes. Now that's different if you've got 10 million at risk or 100 million at risk. So you're definitely going to change your behavior if you've got more financial exposure. But the point is then, so using this very normal part of a fund structure, we're able to use this now strategically to do some really to de-risk some really early, very exciting but really early projects. So we've actually been using this in that way for a few years now. One of the things that happen when we bring stuff in is that it doesn't repeat. So, this is just, sorry, I just have to suck this up. This is what happens. So, somebody will come in. It's a crazy idea. The PowerPoint presentation looks great, but one of the things that we want to do is we want to get those done independently in external labs. Quite a few drop out when we do that. So, it all comes down to I don't think people are being fraudulent. It's just that they're being overly optimistic in their assessment of the data, or they've got a funny machine that always gives really great results. And when you take that outside of that lab, it doesn't look quite so great. But the more important thing is that it basically enables us to get the program moving, work with the scientists and the entrepreneurs, and it reduces our failure rate later on. We know this, that this is basically what we found. And it really only needs one or two winners to come out of the overall approach for it to really make it worth your while. So if I go on, so this is actually quite old, this seed matrix slide, but it actually it's about right. So 7 projects, 3 and a bit million invested. We've written off 3 projects out of that, not necessarily because they don't work, it's just because they're not really interesting enough. And 4 of them remain live. One of the ones that remained live, and actually is becoming more or less the poster child for this approach within Index, is a company called Levicept. So now this company actually exists. And this was a guy out of Pfizer in Sandwich when they closed down the Sandwich site. One of the guys who came out of there had been working on anti-NGF and the analgesia in the analgesia programs there. And he came up with a theory, a hypothesis, that to explain why anti-NGF should be so powerfully analgesic, and anybody who's worked on these programs will know that that's absolutely true. It's incredible. But also why they weren't the right modality for treating pain. So when you've got a damage or injury, you have an increase in a number of neurotrophins, actually, including nerve growth factor. And that's a known. That's not a hypothesis. That is actually fact. Those neurotrophins also cause pain. That's a fact. But Simon's hypothesis is that they're also necessary for repair. So effectively, this is your response. Your body's telling you that there's something wrong by giving you pain, but it's also providing some of the factors to repair that, the underlying damage. And his view was that the anti-NGFs, which and there are several of them, they reduce the nerve growth factor concentration to 0, and that removes the pain, but it also removes the facility for repair. And that there's an alternative approach using a natural receptor for neurotrophins, including nerve growth factor, called P75, would be different in the sense that it would buffer the response, but not ablate it. And that would fundamentally be more balanced, a better approach to treating chronic pain, particularly in osteoarthritis, for which there is basically nothing. So his hypothesis was P75 will be analgesic, but will allow repair. Okay, so that's a hypothesis. It's a twinkle in his eye. How do we turn that into something real, something that we can evaluate and decide what to do with next? So what we designed with him was a killer experiment. And this is basically a non-clinical, you have to bear with me, we can't do all of these experiments in humans. So this was done in a rat model. And what we did was we adapted an existing model of OA in rats induced by an agent called MIA, but we wound it back a bit. The conventional model is actually quite aggressive, so it basically causes destruction of the joint over just a few weeks. It's very severe. We dialed that back to make a much milder form. It's still rapid in the context of human disease, but it develops over 12 weeks as opposed to over 2 or 3. So we developed that model. We tested whether an anti-NGF would relieve pain in that model, which it does, and whether or not it would exacerbate OA, because that would be the consequence of that hypothesis. It would be the prediction. And if that was the case, and we showed that, then does P75 do anything different from the anti-NGFs? And that's a go, no-go experiment. So we agree this is what we're going to do. We agree that this is a success criterion. If it works, we continue. If it doesn't, we wrap it up and we walk away friends. And so that was basically agreed up front. The outcome, so both of these molecules are analgesic, but the actual outcome of the killer experiment was a histology. I'm not going to go into this in any detail, but I'll just point out, because it's actually quite neat. So the panel, that's rubbish. The panel A is a normal joint. So, and it's been stained for the proteoglycan layer. So what you can see on your left, in the top left-hand panel, is the growth plate, and then the cartilage is on the outside of that. So basically it's staining cartilage, but it also stains the growth plate. B is a joint that's been treated with MIA. So you can see that the cartilage layer is obviously much thinner. You're starting to see some destruction of the bone. The foamy stuff, the foamy black stuff is bone marrow. It's basically a damaged joint. But most importantly, in this view of it, the cartilage layer is much reduced. C, at the bottom left, is those same animals treated with MIA that have also been treated with anti-NGF. And in fact, it's made it worse. So in fact, his prediction was correct. It has, in this model, made the pathology worse, and there's very little cartilage to be seen. The surprising thing for us was that D is the same animals or the same experiment treated with P75. And not only is the damage not as great as with anti-NGF, in fact, it's closer to the MIA. In fact, it looks as though there is more collagen there, but this is a 12-week outcome. So there's a bit more diffuse cartilage there, but it's better than we expected. So this actually turned out better than we expected. So there's something interesting happening here with regard to repair of the pathology. So this might actually be not apart from being analgesic, it might also be disease modifying. It's certainly, and if that's the case, that's a really big deal in OA. If you've got analgesia and disease modifying label in OA, that's a very big deal. So in fact, that was enough, and if we did a Series A financing on the back of this particular slide to now test that. So we're basically going to take that all the way through to clinical development and test it in OA patients. But that was done with £750,000, that particular experiment. So that was quite an expensive killer experiment, but it was definitely worth doing. So we'll see how that one plays out. But that's one example of this approach. Another example, the second example of this, is a company called XO1, which is a company we put together in Cambridge, and this is about anticoagulation. And it comes from a very interesting source. So the field is really one that sits on a balance, on a seesaw, where you're trying to anticoagulate the patient so they have a lower risk of thrombosis and heart attacks. But at the other side, the more you do that, the more you expose them to risk of bleeding. Right? So the big problem with anticoagulants is the bleeding risk. It has to be managed in all of them, and that seesaw, that balance, hasn't ever really been nailed, despite a huge amount of effort being put on it. But in fact, the observations that were made in Cambridge suggest that connection, that link, can in fact be broken, so that you can get improved anticoagulation without having a concomitant increased risk of bleeding. And it came from a patient who walked into the A&E department in Addenbrooke's in Cambridge, having fallen in her kitchen. So this is a 54-year-old lady. She fell in her kitchen, she whacked her head really quite badly, and she presented in A&E with a subdural hematoma. So basically the side of her head was out here, and they were prepping her for surgery. The CT scan confirmed that she'd got a subdural hematoma, so the intention was to basically drain it, remove the clot. And as part of that, then they send the bloods down to hematology and just to check her out for her clotting characteristics. And it turned out she was completely anticoagulated, like she's on an anticoagulant. So her consultant said, no, she's not under any anticoagulation therapy. She's not on warfarin, she's not on anything else. But in fact, her blood showed that she was completely anticoagulated. And looking into her history, she should be a bleeder, right? She's not. By any measure, she's not a bleeder. So she's completely anticoagulated, but she's not a bleeder. So this is a really interesting patient phenotype. And it just so happens that she landed in the right place because there's quite a big hematology and clotting research group in Addenbrooke's, and they were on this like a rash. So they went in and took a look at what was responsible for that, and it turns out she's got an autoantibody to thrombin, and it's a particular part of thrombin. So this is just a naturally occurring autoantibody. They often occur later in life. You get these like odd antibody specificities that sprout up, and they can be there for a lot. I mean, she's got 3 grams a litre of this stuff, right? And anybody over the age of 50 probably has 2 or 3 of these floating around. Most of the time they're completely null phenotypes, but occasionally something interesting happens, and this is one of those cases. And we could prove this because we took the antithrombin specificity, we put it into various model systems, in vitro and animal systems, and we could prove that the activity went with the antibody. So the antibody is actually properly responsible for this phenotype. And in fact, so the experiments we did with this when we came into it were largely confirmatory that the properties of the patient were recapitulated with just the antibody from her serum. We manufactured another similar antibody to show that it was actually this specificity that was necessary and sufficient, and it was. And from that point on, then we went in and we've done that in full scale. So that's now on track into the clinic. It's not, the interesting thing is that the patient, it seems that she's got no bleeding risk despite 3 grams a litre in her blood. You can actually make rats bleed if you put in enough of this stuff, but it's about 3 orders of magnitude. The difference between rats being anticoagulated and rats being exposed to increased bleeding risk. And rats are very, very sensitive to bleed. So this looks like it has indeed broken that link between coagulation and bleeding risk. So therefore, it is definitely a very interesting innovation. So why is it compelling? It's basically a huge and important market. I mean, I don't know how many of you work in that area, but it's an enormous market. And no game changers, despite massive effort. And actually, even though there's only one patient who's got this stuff, the fact that you've seen what this does in a human setting is actually quite reassuring that you're not barking up the wrong tree. So one of the questions I was asked to cover is where's pharma in all of this? And the answer is right alongside us. So one of the reasons I've hung out with Index for so long is we like doing experiments. Everybody is up for trying something new, trying something different. And that means every part of your day-to-day life, including how you structure a venture capital fund. So pharma inside venture capital funds is nothing new. There are lots of them. There might be people at this conference. This is slightly different from the usual flavor in that we've got 2 pharma companies in alongside us. We're not actually just an arm of SR1 or JJDC. These are the corporate entities in alongside us, and they have no hard rights and no decision making. So you'd have to ask, why are they there? What are they in it for? Because for us, this works really well. We're able to tap into the brain trust of 2 of the largest farmers on the planet, and for us, that's just fantastic news. I mean, we're just much more well informed now in terms of the things that we're looking at than we have been in the past. What they get out of it is that actually they get full exposure to these companies as they develop. So they regularly meet up with the companies. They regularly get progress updates. They get involved in how these programs are shaped. And if there's something there that they really like, then they're in pole position. They've grown up with these companies. They know them really well, so they know exactly what it is and what it isn't. And which, if you're presented with something from the outside, if JP Morgan ring you up and say, look, I've got a great company for you here, then you have, and you have 3 weeks to make a decision on it. That's a completely different dynamic. So, so far, this is an experiment that's working out very, very well. I think it's going to get replicated in other funds. It'll certainly get replicated in ours. And it's turning out to be a very, very interesting journey. So in terms of the financing the innovation part of my objective, I always remember a saying from a guy I knew who used to work at Merck, is that it's not the smartest who survive, it's the most adaptable. And I think that's probably, it sounds a bit glib, but I think that is absolutely true. We just have to adapt. And it's remarkable how difficult it seems to be for some to adapt to a changing environment. And one of the things that we're seeing, and it's quite obviously a theme of this conference, is there's just an unprecedented level of integration these days. So we are actually becoming much more connected as different satellites within this universe. So everything is partnerships. In fact, I signed a deal last year, I think it was, with Lonza to manufacture all our biologics. So all of our portfolio companies that are manufacturing biologics, we've got a template agreement with Lonza. Lonza manufacture them all. So rather than being some tin pot company coming along and starting from scratch every time in discussing with Lonza how we're going to do this, then we have that more or less nailed already, and we just get things much more slick in terms of moving these programs forward. But especially with the pharma guys, we're learning a lot from one another, actually. So just at how the VCs go about doing things, how we go about de-risking programs versus their approach and their knowledge, which is way more deep than ours. We can't. There are only a handful of us. We can't be masters of everything. And so from our perspective, we're just going to keep doing the experiments, and I think that's probably the right thing for all of us to do. If it's just work out how many rules you have to bend to get something new done, and go off and bend them. Thank you. Yeah, if anybody's got a question.
David Davis, Futura Medical. I'm a very big fan of the virtual model. I think it's terrific. Given the nature of the audience, though, I just wondered if you'd like to comment on the fact that financiers see the transaction between a sponsor company and a CRO as being purely transactional. You pay the money, you get the outcomes. It's a very simplistic view, of course, and many of us struggle with making that interaction work. So from your point of view, do you want to make a comment about things like delays during clinical trial progress and what investor sentiment is around that?
Right, it's a really good point. I mean, and I would have to say that we don't look at it that way. So this is a partnership, because basically if we fail, so if we as the financier fail, or you as a CRO fail, we just failed, okay? It's a question of where the finger gets pointed, but it doesn't really change the outcome at all. So we're basically, with Lonza, it's much clearer. I just put that out there because it just reflects our approach to, and how it should be done, because it is a partnership. There isn't, on the second point, there is an expectation that you are going to get delays. I mean, there are two things that rate limit small biotech companies. One's manufacture of the drug substance, and the second is recruitment in the clinical studies. Right? Those are the two things that rate limit it. Nothing else should be rate limiting. When it comes to delays, some of those are inevitable. They're just going to happen. Right? I can't think of a single program where we haven't hit delays somewhere along the line. Okay, so you can either whinge about that, or you can actually make it okay. And one of the things about the virtual model is that it makes it okay, because the big cost, the reason financiers are going to get pissed if there are delays, is because you've got a cash burn. So if you've got 70 people that are all burning cash while you've got a 6-month delay, that's 6 months times 70 people worth of cash that's just evaporated. But if you've got, like for example, XO1 is 2.5 people, Levicept is 1.2 people, right? The cost of a delay is tolerable, right? So I think you've just got to make it okay. You know, this is the real world. Stuff like this happens. You try and avoid it happening, but it does. You're always breaking into new ground. So you just got to make it okay. So I think with more and more VCs are becoming fans of the virtual model. It's harder work. That's why they're not they weren't immediately a fan base. There's a lot of hard work with a virtual company, because you can't just say to somebody in the management team, can you go and look at this? If you want to look at this, you pick up pick a computer and you go and look at this. So it's much harder work, but it's much more capital efficient, and the cost of delays is much reduced. It's not zero, but it's tolerable.
Anna Matranga, Ipsen. Thanks very much for a very interesting presentation. What I'd like to understand is how do you make sure that it's really a killer experiment, that you're not going to invest more money because maybe you missed something?
Yeah, it's a good question. We don't know for sure. What we try and do is triangulate that as much as we can. So this is actually where the pharma and other experts, the due diligence on an early stage program, most of it's a waste of time. The thing that's not a waste of time is, okay, what do we really need to know? What really is going to be the critical point that turns this from a, interesting but we don't know project to one where we can say, yes, definitely, if it does this, we are then interested. Probably the pharma guys have the biggest say in this, because they've probably seen something like this before, or they've got programs that, or they've had programs in the past that are in this area, and they say, oh no, well, we've had a load of programs in this area, but they've all failed for this reason. So that tells you, if something new is coming up in that same area, it has to succeed on that particular point where everything else has failed before. But it's a stepwise thing. So you don't try and get it all done in one fell swoop. It would be very complicated. But we try to reduce it to a binary outcome. I think that's probably the mindset that we go into this with. But the short answer is we don't know. All we can do is design something that says, if it does this, then we are going to be interested in it. We are going to be able to interest other people in it, and we'll get to the next step. But it's really trying to get the discharge as much of the risk or the uncertainty with a small amount of money, and not particularly a short amount of time. Actually, we're not so time conscious on these things. I'd rather it was done once, done properly, and all we'll commit to is that we'll put the best we can. We'll give this the best shot. So you decide what it is, give it the best shot. Good example, the Leviset program. We manufactured the anti-NGF and the P75, as though we were developing this for clinical study. So it's not to GMP, but it's not a Saturday afternoon fermentation either. So if you get the answer, you have to be able to live or die by that answer. But you're right. I mean, it is, at the end of the day, it's a judgment call. You just try and be as best informed as you can.
Okay, thank you.
Yeah, thanks very much.